Welcome to this explainer. Right off the bat, I want you to totally shift how you think about the human body. Don't just picture flesh and bone, okay? Think of it as a vast, incredibly complex biological system. Imagine billions of cells communicating in absolute perfect harmony, and all of it is driven by an intricate biological code. To put that into perspective, your DNA contains over 3 billion letters of code. That's a literal cosmic flow of data happening inside you right this very second. But what happens when there's a glitch in that system? What happens when a piece of that biological code gets corrupted and actually starts causing damage? Well, today, we're going to look at one of the absolutely most astonishing breakthroughs in modern medicine. We're going to explore how scientists are learning to literally read our underlying code, identify those exact glitches, and reprogram our bodies to fight back. Okay, let's dive into this, because it sounds like a question ripped straight out of a sci-fi movie. Can you actually vaccinate against your own cancer? For decades, we've thought of vaccines as just something we get to stop an outside virus like the flu from making us sick. But the breakthrough we're exploring today, it completely flips that entire concept on its head. We aren't talking about some generic vaccine made in a factory and shipped out to millions of people. No way. We're talking about a highly personalized, custom-built defense mechanism that targets a disease you already have. It is a stunning reality where medical science is literally treating the body as an operating system, one that can be patched and updated. So, how does this actually work? Well, the whole process kicks off in the operating room and the lab. When a patient has a tumor, the very first step is to physically remove that cancer. But here's where it gets wild. Instead of tossing that tumor into a biohazard bin, they rush it over to a specialized lab. And it doesn't go alone. It's sent right alongside a simple tube of the patient's normal, healthy blood. Think of this as setting up a massive technological comparison. On one side, you've got the clean code. That's your normal blood, representing your body's baseline healthy programming. And on the other side, you have the corrupted code, the removed cancer. By putting these two side by side, scientists get the exact materials they need to launch an incredible biological investigation. Once those samples hit the lab, the place basically turns into a futuristic data mining operation. Scientists use some seriously cutting-edge technology to sequence the genome of both samples. They aren't just looking at these cells under a microscope anymore. They're actually reading the fundamental source code. They literally download the entire biological operating system of both the healthy cells and the cancerous cells, so they can sit down and read them, line by line. Now, what's really interesting about this slide is the sheer mind-boggling scale of the data they're processing here. I mean, think about this. Sequencing the human genome means analyzing between 20,000 to 30,000 unique genes. 30,000! Imagine someone asking you to spot a single tiny typo hidden inside 30,000 highly complex instruction manuals. That's what we're talking about. The technology has to scan through every single one of those genes in the healthy cells, and then turn around and do the exact same thing for the cancer cells. It's a massive, massive undertaking of data extraction and comparison. We're talking about finding a microscopic needle in the world's biggest biological haystack. The pipeline to actually pull this off is just incredibly elegant. First, you sequence that massive cancer genome right alongside the normal DNA. And because both of these samples came from the exact same person, their baseline code should be completely identical, right? It's kind of like running a file comparison on your computer. Any differences, any anomalies that pop up only in the cancer sequence, those are the biological bugs causing the disease. By isolating those specific tiny bugs, scientists can move on to the final, most crucial step: actually synthesizing a cure. So, the crucial point is that by running this super precise comparison, all the background biological noise is completely filtered out. As researchers put it, you are left with the smoking guns of only the cancer mutations. You aren't just guessing what might be causing the cancer anymore, and you certainly aren't using some broad, toxic treatment that's going to damage healthy tissue in the crossfire. No, you have a precise, fully mapped out list of the exact genetic glitches that are totally unique to that specific person's tumor. The bugs in the system have been completely exposed. Let's move to and see how this builds, because now we have to ask, how do we take this from a digital screen back into the physical world? Well, once scientists have the data identifying those specific mutations, they do something truly, absolutely extraordinary. They literally print out the protein of the mutations. And I'm not talking about a 3D printed plastic toy here. Using the sequence data as a blueprint, they use advanced lab techniques to physically manufacture custom biological material that perfectly corresponds to those isolated cancer mutations. It is pure sci-fi turned into everyday medical reality. And this brilliantly illustrates the final piece to the puzzle. That freshly printed protein is then injected right under your skin. Because it mimics the cancer's exact mutations, but importantly, it isn't the dangerous cancer itself, it acts basically like a wanted poster for your immune system. Your body recognizes this foreign, corrupted code, mounts a highly specific defense, and learns exactly how to destroy it. You have essentially just uploaded a custom antivirus patch directly into your immune system. You are quite literally vaccinating yourself against your own cancer. How amazing is that?
As we wrap up this explainer, I really want to leave you with this thought. We have just walked through a process where we literally extract a disease, read its source code, print its opposite, and use it to program the human immune system to fight back. We are no longer just treating the body. We are treating it as a programmable system. If we can sequence a genome of 30,000 genes to find the smoking gun of a cancer and physically print proteins to fight our own mutations, what else is possible? The horizon of personalized medicine isn't just approaching, it is already here. Keep questioning, keep learning, and really think about the vast, incredible potential hidden right there in your very own biological code.